Bismillahirrahmanirrahim. In this lecture, we will discuss about the topic of the endocrine and excretion of pancreas. Basically, the pancreas is made up of four types of cells, which is alpha cells, beta cells, gamma cells, and Fc cells, and Pp cells. Then these all type of cells will secrete the different type of secretions. That the alpha cell will secrete the glucagon, beta cell secrete the insulin, gamma cell will secrete the somatostatin, and the Fc और PC cells will secrete the pancreatic polypeptide. So हमारे पास क्या है कि जो पेंक्रियास है, पेंक्रियास इज अ डुअल ग्लैंड. पेंक्रियास की एक्सोक्राइन सिक्रीशन भी होती है, पेंक्रियास की एंडोक्राइन सिक्रीशन भी होती है. सो पेंक्रियास एक्सोक्राइन सिक्रीशन जो है, जो कि एक्सोक्राइन ग्लैंड टेक्निन के लिए अपनी सिक्रीशन जो है इसमें देते हैं. दे कैन सिक्रीट देयर सिक्रीशंस इन द डक्ट. ठीक. तो ये एक्सोक्राइन सिक्रीशन जो है वो हिपेटोपेंक्रियाटिक डक्ट के थ्रू, हिपेटोपेंक्रियाटिक जो है वो डक्ट के थ्रू जो है वो स्मॉल इंटेस्टाइन में जो है वो ओपन होती है. So that was all about the exocrine secretion, and the endocrine secretion is basically different type of cells which are present in the pancreas, which secrete their secretion directly into the blood. So these type of cells are the alpha cell, beta cell, gamma cell, and Fc cells is the Pp cell. So the first and the main hormone which is produced is the beta cells. From the beta cells is the insulin, and insulin the secretion source is beta cells, and the chemistry is the polypeptide chain which is composed of 51 amino acids. That means insulin is basically made up of 51 amino acids. In the 51 amino acid, 21 amino acid chain is the alpha chain, and 30 amino acid chain is the beta chain, and these two chains are attached with each other through a disulfide bridge. So the biological half-life of insulin is only 5 minutes. That means in the 5 minutes, the insulin will be degraded after performing their action, and after completing their half-life. If it's not performing their action, then the insulin will be degraded through the liver and the kidney. So insulin plasma level normally is 10 microunit per ml. And synthesis of insulin. If we talk about the synthesis of the insulin, insulin is mostly synthesized in the pancreatic beta cells, first as a pre-proinsulin, then proinsulin, and then insulin. So these pre-proinsulin insulin are from the messenger RNA. When the insulin messenger RNA is produced, so the transcription and the translation of the messenger RNA is taking place. Then in the ribosomes and endoplasmic reticulum, the pre-proinsulin is converted into the proinsulin, and proinsulin is then converted into the insulin. So you can see here at different type of cleavage of polypeptide chain and amino acid will be take place in the endoplasmic reticulum during the process of packing in the Golgi apparatus also, and then this cleavage and process will complete it. Then the end product is basically the mature insulin, which is 5-carboxy polypeptide E product produces the mature insulin. So in this diagram, you can also see here there is the alpha and the beta chains which are attached to each other, and different type of cutting procedure will be take place here, and finally the insulin formation will complete it. Then what are basically the action of the insulin on the insulin perform different type of function on the carbohydrate, on the fat, and on the protein. So on the carbohydrate, basically it will cause the increase in the transport and the uptake of glucose by the cell, promote the peripheral utilization of the glucose. That means it will cause the entry of the glucose into the cell and promote the storage of the glucose in the form of glycogenesis in the liver. This mechanism will be take place, and inhibition of the glucogenolysis. That means the cut down of glycogen into the glucose and breakdown of the glycogen into the glucose, and inhibit the gluconeogenesis, the formation of the glucose from the other products, non-carbohydrate products, you can say also. So on the carbohydrate metabolism increase and transport of the uptake of the glucose, it basically increase the permeability of cell membrane of the glucose. When the permeability of the cell membrane of the cell will increase, it will facilitate the transport of the glucose into the cell. So insulin also increase the glucose transported protein, that is glucose transporter protein 4 in the cell membrane. This glucose transporter protein then cause the increase in the transport of the glucose into the cell. In this diagram, you can see here, when the insulin attached to the receptor, then it will cause a signal transduction cascade, and this will cause the glucose transport 4, and this glucose transport 4 is basically exocytosis. Exocytosis, that means it will be exported out of the cell, and when it is exported out of the cell, it will cause a different opening of the ion channel. Ion channel you can not say, you can say the glucose channel will be open here. When the glucose channel and glucose transport, you can say glucose transporters number increases in the cell. So on the carbohydrate, it will cause the peripheral utilization of the glucose. That means in the presence of the insulin, glucose which enters the cell is oxidized immediately, and the rate of the utilization depends upon the intake of the glucose. So glucose is entered into the cell in the presence of the insulin. It causes the promote of the storage of the glucose, that is glucogenesis. Glucogenesis insulin promotes the rapid conversion of the glucose into the glycogen, and the glycogen is then stored in the muscle ending. It also inhibits the glucogenolysis. Glucogenolysis is basically the breakdown of the glycogen into the glucose in the muscles and liver. Inhibit the gluconeogenesis, the formation of the glucose from the non-carbohydrate sources. On the protein metabolism, it will cause all the mechanism that will cause the synthesis and the storage of the protein, as it will cause the synthesis and the storage of the glucose in the form of glycogen. It also increases in the formation of the different type of mechanism, which will cause increase in the protein synthesis, that may facilitate the transport of amino acids into the cell. When the transport of amino acid into the cell from the blood is take place, that means there will be the anabolic function of the protein to take place here. It also causes accelerating the protein synthesis by influencing the transcription and translation. When the transcription and the translation process will fastly going, then the protein synthesis procedure also will be accelerated, and then preventing the protein catabolism. The protein catabolism, because insulin is performing the anabolic function, so inhibit the catabolic function. It also causes the conversion of the protein into the glucose. Protein is also converted into the glucose through the different type of cycles, which are discussed in biochemistry. So these cycles will cause the conversion of the protein into the glucose. So because insulin also has the anabolic function of the protein, so it will prevent the formation of the protein from the production of the glucose, conversion of the glucose, conversion of the protein into the glucose. On the fat metabolism, you can say the synthesis of fatty acid and triglyceride. On the same, in the form of fat, it will also cause the storage function of the fat. The synthetic function of the fat. The synthesis of the fatty acid and triglyceride, transport of fatty acid into the adipose tissue, and storage of fat. On the growth, insulin also has a promotion of the growth of the body, that is basically the anabolic function. Mode of action of the insulin. The insulin is basically attached to the receptor, and the insulin receptor complex will form that will cause the activation of the intracellular enzyme. And when the intracellular enzyme are activated, different type of functions of the cell will be performed by the cell. Then regulation of the insulin. Insulin secretion is mainly regulated by the blood glucose level. The blood glucose level will cause mainly the regulation of the insulin, the regulation of the insulin secretion. When the blood glucose level is low, the insulin secretion is low. When the blood glucose level rises, the rate of the insulin secretion will also rise in. And when the blood glucose level is above the 200 milligram per deciliter, the rate of the insulin secretion will also rise rapidly to 400 milligram micro unit per minute. That means the increase in the level of the blood glucose will cause the increase in the secretion of the insulin from the pancreatic beta cells. Then there is a biphasic effect of the glucose. What is biphasic? First of all, we discuss about the role of the insulin on the carbohydrate, proteins, fat. After that, we are discussing about the regulation of the insulin, how the insulin regulate the blood, how insulin is basically regulated. It is regulated by the glucose level in the blood. The glucose level in the blood when low, the insulin level will be low. When the glucose level will high, the insulin level automatically high. Then there is the biphasic effect. Biphasic effect of the glucose is basically when initially when we eat meal, then there is a sudden rise in the insulin level. Then after that, the insulin level will fall and steadily and you can say it has reached toward the peak with a sustained condition, and these two peaks level is known as the biphasic effect of the glucose. So in this diagram, in this slide, you can see here, when the blood glucose level increases after a meal, the release of the insulin in the blood increases. That means, as the blood glucose level in the blood after meal increases, the insulin level in the blood will increase. That is the first piece of the biphasic effect of glucose. And when few minute concentration of the insulin in the plasma increases up to 100 microunit per ml from the basal level, that is 10 microunit per ml. The basal level is basically the normal level of the, you can say the normal level of the insulin in the blood. So it is because of the release of the insulin that is stored in the pancreas. Because insulin is stored in the pancreas, when there is signal comes, these insulin that is uh, uh, stored in the vesicles, these vesicles attached to the membrane and rest will occur there, and the beta cell will secrete the insulin. So first we have the stored insulin in the beta cell. So when the glucose level in the blood rises, the stored insulin from the pancreatic cell is released into the blood. But as the blood glucose level is more high, so there is sudden fall in the insulin graph, you will say. This fall is due to stored insulin deficiency, because the stored insulin is mostly used. Then after 10 to 15 minutes, the blood insulin concentration in the blood is reduces, and that is 40 to 50 microunit per ml. So the basal rate that was 10 microunit per ml, then there is increase, that is 100 microunit per ml, then there is decrease, that is 40 to 50 microunit per ml. Then after 15 to 20 minutes, the insulin secretion arises once again, and it will reach about 2.5 hours. So after meal, you can say the biphasic effect is that in which the insulin level will go toward the peak situation, and this peak will come two times. The one is directly after the meal, and the second after 2.5 hours. And after the 2.5 hours, there is certainly, because production of the insulin, again production of the insulin will take place in the pancreatic beta cell, and this will take time now. The insulin in the insulin secretion, when this will take time from the secretion of the insulin, then there is 2.5 hours, uh, and after 2.5 hours, there will be again peak in the secretion of the insulin. So these two peaks which appear at different times, first after meal and second after 2.5 meals, is known as the biphasic effect of the insulin. So regulation of the insulin secretion is also through the role of the protein. When there is excess amino acid in the blood, that will cause the insulin secretion stimulate. Why? Why? Because amino acid is stored in the muscles and in the liver. Okay, so when the amino acid level in the blood is increased, the pancreas will secrete the insulin, and this insulin then cause the entry of the amino acid into the cell and stored in the cell. So the most potent amino acid is arginine and lysine. So arginine and lysine are the two major amino acids which mostly cause the secretion of the insulin. The third role is the regulation is the lipid derivative. You can say all the actions which are performed by the insulin on the carbohydrate, on the protein, on the fat will cause the regulation of insulin also. So the beta keto acid such as the acetoacetate also increase the insulin secretion. So when the beta keto acid is present, it will cause the insulin secretion. There are different type of hormones that are secreted from the gastrointestinal tract will also cause increase in the secretion of the insulin. That is cholecystokinin and gastrointestinal inhibitory protein. Then there is the role of the endocrine hormone. The endocrine hormone will also regulate the secretion of the insulin. The glucagon, growth hormone, and cortisol also stimulate the insulin secretion indirectly. There is different type of autonomic nerves. Then autonomic nerve will also cause the regulation of the insulin. The autonomic nervous system, the parasympathetic nervous system, will cause the increase in the secretion of the insulin, and the sympathetic system, which is basically the fight and the flight system of the nervous system, will cause inhibition of the secretion of the insulin. So the role of the nerves is also unclear, but they have the role on the insulin. So first we discuss about different type of cells of the pancreas, then different type of then there is the insulin production, which is through the beta cell, and this beta cell will perform beta cell will basically secrete the insulin, and then this insulin perform different type of functions of the carbohydrate, protein, and the fat. Then there is the regulation of the insulin, the biphasic phase, and how the insulin is regulated to different type of carbohydrate, protein, fat, glucose level in the blood, and different type of endocrine hormones and growth hormone. So the second hormone which is secreted from the pancreas is the glucagon, and the glucagon has also secreted from the alpha cells of the eyelids of the Langerhans, and the glucagon is also a polypeptide, and the half-life is brief. Half-life of the insulin was 5 minutes. Synthesis is also as a pre-proglucagon, then proglucagon and glucagon, and the metabolism is it is degraded in the liver and the kidney. Regulation of the glucagon secretion is controlled mainly by the glucose and the amino acid level in the blood. So, the role of the blood glucose level, the important factor that regulates the blood secretion of the glucagon is basically the blood glucose level. When the blood glucose level decreases below the 80 milligram per deciliter, then the more glucagon is secreted from the alpha. And the role of the amino acid, when the amino acid level in the blood increases, it stimulates the secretion of the glucagon. So glucagon in turn convert the amino acid into the glucose. So glucagon function is mainly on the carbohydrate metabolism. Its inhibition of the glucose glycolysis, increasing the gluconeogenesis of the protein. It will cause the glycogen increase, the transport of the amino acid on the fat. It will cause the glucagon show the lipolysis and ketogenic. तो अगर हम इन सारे एक्शंस को जो है वो देखते हैं, तो ये सारी जितनी भी एक्शन है, जैसे कि कार्बोहाइड्रेट के ऊपर ग्लाइकोजेनोलाइसिस को और ग्लूकोनियोजेनेसिस को, ग्लूकागॉन इज इंक्रीजिंग बट इंसुलिन वाज डिक्रीजिंग। इसी तरह से प्रोटीन मेटाबॉलिज्म में अब ये प्रोटीन को जो है वो ट्रांसपोर्ट कर रहा है अमाइनो एसिड को इनटू द लिवर सेल, और यू कैन से द अमाइनो एसिड आर यूटिलाइज्ड फॉर द ग्लूकोनियोजेनेसिस. And the gluconeogenesis was a process which is the formation of the glucose from the non-carbohydrate source that may be in the form of protein, but insulin was inhibiting this procedure. On the fat metabolism, it will show the lipolytic and ketogenic action, while insulin was showing the storage function of the fat. So these all function will show that these are totally opposite to the insulin. Then there is the role of regulation of the glucagon of the secretion, the role of the blood role of the amino acid we already discussed. Then there is the somatostatin, that is third hormone of the pancreas, that is secreted from the delta cell in the eyelids of the Langerhans, and the delta cell in the stomach and the upper part of the small intestine, and hypothalamus. The chemistry of the somatostatin is polypeptide. Okay, that was looking like the gamma cells, but not. It is the delta cell of the pancreas. So action of the somatostatin basically inhibits the secretion of the both glucagon and the insulin. So it reduces the secretion of the gastrointestinal hormone. Somatostatin inhibits the secretion of the growth hormone and TSH from the GLP-2. This is why it is known as the growth hormone inhibiting hormone. So pancreatic polypeptide is also secreted away from the islets of the Langerhans, which is the fourth hormone, but the chemistry is polypeptide in nature. But the exact physiological action of the pancreatic polypeptide is yet not known. Basically, it increases secretion of the glucagon from the alpha cell in the islets of the Langerhans. Then the applied physiology of the insulin, that is high pancreas. The applied physiology will cause the hypo and the hypersecretion. The hypersecretion will cause the diabetes mellitus, and the hypersecretion will cause the hyperinsulinism. Hypoactivity will cause the diabetes, and the diabetes is developed is primary and secondary. The primary diabetes is related to not with other disease. You can say it will not associate with other disease, while the secondary diabetes will associate it with the other disease on other damage. Then the primary diabetes is further divided into two, type 1 and type 2 diabetes. Type 1 diabetes is basically deficiency of the insulin because of the destruction of the beta cell in the islets of the nectar. So it may occur at any age, but it usually occur before 40 age. And so it totally depends on the insulin. So it is known as the insulin dependent diabetes mellitus and type 1 diabetes mellitus. And when it develops at infancy or the childhood, it is known as the juvenile diabetes. So what are basically the causes of the type 1 diabetes? Degeneration of the beta cell, destruction of the beta cell, congenital disorder of the beta cell, and destruction of the beta cell during autoimmune disease. So when there is total destruction of the beta cell of the pancreas due to different reasons, then the secretion of hormone will not occur there. So this type of you can say if the diabetes is known as a diabetes, insulin dependent diabetes, because the patient is unable to produce insulin due to destruction of the beta cell. So, in type 1 diabetes will also relate to the latent autoimmune diabetes in adults and the maturity onset diabetes. The latent autoimmune diabetes is slowly progress, and maturity may be rare, inherited, and from diabetes that occurs before 25 years of age. Type 2 diabetes mellitus. Type 2 diabetes mellitus is basically due to insulin resistance and failure of the insulin receptor to give response to the insulin. So it is insulin is produced, but the receptors are unable to recognize insulin and to attach with the insulin, and this is known as the receptor defect. It occurs after 40 years of age, and it is also known as the non-insulin dependent diabetes. Because the non-insulin dependent, we can say it also because different types of medicine given to the patient can also manage the insulin level in the blood. So type 2 diabetes mellitus has a genetic effects in lifestyle changes and stresses. Other form of type 2 diabetes mellitus is the gestational diabetes and pre-diabetes. Gestational diabetes mostly occur at the time of pregnancy, and pre-diabetes mostly also called chemical, subclinical, latent, and borderline diabetes. And it is a stage between the normal condition and diabetes. There is the difference between the type 1 and type 2 diabetes. Type 1 diabetes and type 2 diabetes at the level of age of onset, major cause, insulin deficiency, and immune destruction. So what are basically the difference between type 1 and type 2 diabetes? We have to know. Secondary diabetes mellitus is basically related to another disease, and it has 2% in presence, you can say. And the causes of secondary diabetes mellitus is due to gigantism, acromegaly Cushing syndrome, due to an endocrine disorder, due to damage to the pancreas, or due to the pancreatectomy, This is known as the surgical removal of the pancreas. What are basically the signs and symptoms of the diabetes? There are different types of signs and symptoms of diabetes: increase in the blood glucose level, mobilization of the fat in the blood, diffusion of the protein in the blood, glycosuria. Glycosuria is loss of. These are basically the sign and symptoms. The first is the glycosuria is basically the loss of the glucose from the urine. Osmotic diuretic. Osmotic diuretic is the diuresis which is caused by the osmotic effect. It leads to the polyuria and polydipsia. Polyuria is increase in the urine, and polydipsia is increase urine void of, and polydipsia is the increase in the water intake. Polyphagia. Polyphagia means intake of increased amount of food, or asthenia is the loss of the strength. Acidosis, because the fat is mostly cut down, and it causes release in the fatty acid and ketone acid, and this will cause the keto acid will be excreted to the urine, and ketonuria will be occur. Acetone breathing is basically known as due to the expired and the inspired breath order, that is in the form of fruity disorder due to the ketoacidic presence. And the small breathing. The small breathing is the increase in the rate of the depth of the respiration. You can say there is deep inhalation and deep exhalation. Then circulatory shock, asthmatic diuretic lead to dehydration and may lead to the circulatory shock. And different conditions will accumulate with each other and cause coma condition when there is deficiency in the insulin, and the glucose level will also be decreased, because diabetic patient may mostly facing a high level and a low level of the glucose at the same time. So the glucose level should be maintained, and if the glucose level is low, and because increase in the glucose level will also cause the hyperosmolarity, and hyperosmolarity that means there is a different level of osmolarity will not be managed in the blood, so it will cause the condition of coma. There are complications of the diabetes mellitus, that is hypertension and the myocardial infarction, and diabetic retinopathy, diabetic nephropathy, and diabetic mellitus. So diabetic patient mostly have the eye, I have the kidney, and have the neural problem. The treatment of the diabetes mellitus is the exogenous insulin secretagogues, insulin sensitizer, alpha-glucoside inhibitor, and diagnostic tests for the fasting blood glucose level and the postprandial blood glucose level, and glucose tolerance test, and glycosylated hemoglobin. What are basically cause of the hyperinsulinemia? This is the hyperactivity of the insulin secretion, that is due to the tumor of the beta cells, and the sign and symptoms of the hyperinsulinemia is the hypoglycemia, tonic convulsions, unconsciousness, and coma. So the assignment of this whole lecture is what is basically the glucose tolerance test and how we perform the glucose tolerance test. You should know about what is the glucose tolerance test. This is assignment for the whole class. You have solved that. Okay, guys, hopefully you will understand. In this lecture, we will discuss about the pancreatic secretion and different type of pancreatic cell and different type of hormone which was secreted from the pancreas, and different type of hyper and the hyper related secretion related disease, the hyper secretion related disease, that were diabetes, their type, their management, their test. So hopefully you will understand in this lecture. Okay, guys, Allah Hafiz.